Hello friends, today we will deal with one of the very important concept in microbiology, sterilization techniques. It is a part of unit 3 in paper 2, that is fundamentals of microbiology. We will see in detail different techniques used for sterilization. First, let us try to understand what is meant by sterilization. So it is the killing or removal of microorganisms from object or surface. Sometimes it is necessary to kill microbes from certain places or to remove microbes from certain surfaces. So this process of removal or killing is called as sterilization. So during sterilization, these microorganisms are subjected or exposed to different antimicrobial agents, maybe physical agents or chemical agents or certain conditions which results in their death. So this overall concept is called sterilization, a removal or killing of microbes from object or surface. Now we will see different methods used in sterilization, out of which one of the most frequently used method in laboratories is moist heat sterilization. It is also one of the most effective method. In this method, the agent that acts as a bactericidal agent is steam under pressure. Now, steam under pressure, I will give you best example as our pressure cooker. Pressure cooker works under the same principle as moist heat sterilization. So this moist heat sterilization is usually carried out in a device in a container. It is called an autoclave, where temperature usually is very high, that is around 121 degrees Celsius. So this autoclave, a device works on the principle of moist heat sterilization through the generation of steam under pressure. What happens in these methods? As you heat the water, it results in generation of this steam. This device, autoclave, in which this process is carried out, is completely sealed, so that the steam generated during heating of the water cannot go outside this autoclave. So it results in build-up of the steam, which increases pressure inside the device, inside the autoclave. So increased pressure results in an increased temperature. The steam which is generated in an autoclave is a supersaturated steam, and this supersaturated steam has very high penetration power. The temperature of the steam is around 121 degrees Celsius, and pressure used in an autoclave is around 15 pounds. So as pressure builds up, temperature also increases, and this steam acts as a bactericidal agent. Bactericidal means it literally kills bacterial cells. It kills means what happens, the proteins of the microorganisms, they get coagulated. Proteins are very important structural components of a cell that make up the cell. So that results in denaturation or coagulation of the proteins of the bacterial cells, which ultimately results in the death of the cells. This is the principle behind moist heat sterilization. Now this is the design. We frequently use this device in laboratories for carrying out sterilization of the medium or sterilization of the glasswares. It is very frequently used in microbiology laboratory, and most of you must have performed the experiment by using this autoclave. It consists of a basket. Below that there is a water, then there is a pressure vessel, pressure gauge is there, safety valve is there, thermometer. So initially checking of the water level of this autoclave is very important. Once you check the water level, you can keep your desires material, glasswares or medium inside the autoclave. Once it is switched on, the water will start to heat. That will result in generation of the steam, and since this device is completely closed, the steam cannot go outside. As a result, it will generate high pressure. Pressure increases, temperature also increases. So we need to carry out sterilization at 121 degrees Celsius for around 15 to 20 minutes. Remember this temperature. Sterilization in autoclave is usually carried out at around 120 degrees Celsius for 15 to 20 minutes. The reason behind this fact is that the most heat resistant bacteria, they are supposed to be Bacillus stereothermophilus, and the endospores of this bacterium get killed at 121 degrees Celsius. Endospores of this bacterium, Bacillus stereothermophilus, they get killed at this much temperature, and therefore the temperature pressure used in autoclave is around 121 degrees Celsius for 15 to 20 pounds. So this is the device in which moist state sterilization is carried out. Another method of sterilization is dry heat sterilization. So it is the process of removing microorganisms by applying dry air or moisture free air. The steam which was generated in autoclave was a moist heat, but here the steam generated or heat generated is a dry heat. It means there is complete absence of moisture. So this heat, it is appropriate for moisture sensitive substances. Many substances can be sensitive to moist or presence of water. They cannot be sterilized by an autoclave. So such substances can be sterilized by means of dry heat sterilization. So this dry heat sterilization is based on the principle of conduction of the heat, which is absorbed by the outer surface of an atom and then passwards to the next layer. So entire item reaches the proper temperature needed to achieve sterilization. This is the principle behind dry heat sterilization. So in this method also, the microorganisms result in death, and this death is due to denaturation of the proteins. Denaturation means conversion of folded protein to unfolded protein. So this results in a loss of function of the protein. It also results in lysis of the proteins. So ultimately effect of moist heat as well as dry heat is on the proteins, which are important components of the bacterial cell. Here the temperature used is rather very high than moist heat sterilization. Temperature used is around 160 to 180 degrees Celsius, and the time required is up to 2 hours. So you can compare the relationship between autoclave and moist heat sterilization and dry heat sterilization. In moist heat sterilization, the temperature used is around 121 degrees Celsius for 15 to 20 minutes, and in dry heat sterilization the temperature used is very high, 160 to 80 degrees Celsius for up to 2 hours. The device used for dry heat sterilization is called hot air oven. Like autoclave, in which moist heat sterilization is carried out, the dry heat sterilization method is carried out in a device called hot air oven. So it consists of an insulated chamber that contains a fan for distribution of the heat. Then it contains thermocouples, temperature sensors, shelves, and door locking controls. So just now I told you the temperature time relationship for hot air oven is comparatively very high. Here you can see the design of hot air oven. Uh, it is frequently used in microbiology laboratory. You can see there is a motor blower, then there is a heater, then working room air duct is there, thermal insulation layer is there, then there is a control panel in which you can set the desired temperature and time. Also temperature control system is there. So this hot air oven also results in destruction of endospores of microbes. Remember, both moist heat sterilization and dry heat sterilization both result in destruction of endospores. Endospores are supposed to be extremely resistant to heat, but they are destroyed when they are exposed to moist heat or dry heat sterilization, as in autoclave or in hot air oven. Another method of sterilization. Now the methods which we are discussing, they are mentioned according to your syllabus. They are mentioned in your syllabus. Methods of heat sterilization we are discussing. First method, moist heat sterilization. Second method, dry heat sterilization. The third method of sterilization we are discussing now is pasteurization. So pasteurization is also called sterilization below 100 or heating below 100 degrees Celsius. In both earlier methods, in moist heat and dry heat sterilization, the temperature used for sterilization was above 100 degrees Celsius. But sterilization can still be carried out at temperatures below 100 degrees Celsius. And one of the best method is pasteurization. Now name itself tells that the person behind this concept was Louis Pasteur. It was Louis Pasteur who discovered the method of pasteurization. So he demonstrated the microbial spoilage of wines that could be prevented by short periods of heating. So he was the person firstly who told that the heating below 100 degrees Celsius can also be effective to kill some microorganisms. Of course, not their endospores. But he was the person who founded a solution to prevent spoilage of wines. Even milk is traditionally pasteurized by one of the method called LTH. LTH stands for low temperature heating. In milk industries, milk is not rather sterilized at very high temperature because it may result in loss of nutrient quality of the milk. So heating of the milk below 100 degrees Celsius can be effective to reduce microbial count. It will definitely reduce microbial count. Even it will result in destruction of certain pathogenic microorganisms present in the milk. So temperature used in LTH, that is low temperature heating, is 63 degrees Celsius for 30 minutes. So this is one of the method of pasteurization, low temperature heating, that will reduce microbial count. That will not remove all microorganisms, but that will reduce microbial count. Then another method of pasteurization is HTST, high temperature short time. High temperature short time means pasteurization carried out at around 72 degrees Celsius for 15 seconds. High temperature short time. In earlier method, low temperature heating, temperature was lesser one but time exposure was larger. Here temperature has been increased and time exposure has been reduced, 72 degrees Celsius for 15 seconds. That is in HTST. Remember, as I told you, this is not the actual method of sterilization, but it ensures the destruction of certain disease causing microorganisms which can be frequently present in the milk as a result of contamination. The examples of these pathogenic microbes include Brucella abortus and Mycobacterium tuberculosis. These were frequently found in milk. So these methods of pasteurization, LTH and high temperature short time, will definitely destroy such disease causing microorganisms. So even in industries nowadays, this method is frequently used for pasteurization. Then another method of heat sterilization is called tyndallization. It was John Tyndall who discovered this method. So some media, microbiological media, then solutions and biological materials, not all of them can be exposed to above 100 degrees Celsius because many of them can be sensitive to high temperature. So in such cases, we can use another method of sterilization, that is called fractional sterilization. This fractional sterilization is also called tyndallization. So this method of tyndallization involves heating the material at 100 degrees Celsius for 3 successive days with incubation periods in between. Listen to me very carefully. Heating at 100 degrees Celsius for 3 successive days with incubation periods in between. Means if you are sterilizing the medium, heat today it at 100 degrees Celsius, then keep it in incubation for 24 hours. After 24 hours, again heat at 100 degrees Celsius. After 2 days, again incubate, again expose it to 100 degrees Celsius. So for successive 3 days, the media should be exposed to 100 degrees Celsius. So during this incubation, the significance of this incubation is that during this incubation period, the heat resistant endospores germinate, and these germinated endospores get easily destroyed when they are exposed to 100 degrees Celsius. This is the principle or significance of heating at 3 successive days at 100 degrees Celsius. If spores are present and do not germinate, then material will not be sterilized. If spores are present and they do not germinate, usually what happens when you expose the endospores to temperature like 100 degrees Celsius, it will result in their germination. Or even a provision of nutrients for endospores will result in their germination. But if the spores do not germinate, then it will not result in sterilization, because 100 degrees Celsius is not sufficient to kill endospores. 100 degrees Celsius can kill the vegetative cells. So during this incubation period, it is expected that these endospores have germinated and converted into vegetative cells, and vegetative cells will get easily destroyed when they are exposed to 100 degrees Celsius. Another method of sterilization. Now till now we have discussed methods of heat sterilization. Another method of sterilization, filtration sterilization. This is not the method of heat sterilization, because not all substances can be exposed to high temperature, not to even 50 or 60 or 100 degrees Celsius also. So for such substances, we can use an alternative method called filtration sterilization. So this filtration sterilization uses certain membrane filters. These filters have certain pores, and these pores will help to trap the bacteria and will give you the bacteria free material. Certain type of filters used in microbiology laboratory include a membrane filter. They can be used to trap and concentrate contaminating microorganisms. Then these filters, suppose you want to filter suppose it's water, you can pour water through this membrane filter, and after you can place this membrane filter or filter disc onto the nutrient medium to see how many microorganisms are present in the water. You can count the number of colonies. Even you can count how many microorganisms are present in how many ml of the water. This is the advantage of this membrane filter technique. Another type of filters include high efficiency particulate air filters. These are particularly used in laminar airflow. It is a device frequently used in microbiology laboratories. It is used to remove 99.97% of the particles of around 0.3 micrometer or larger even. Another type of filters include sintered glass filter. They are prepared by size grading powder glass and followed by the heating. And this pore size can also be controlled by the general particle size of the glass powder. They can be easily cleaned and they have low absorption properties and do not shed particles. These are the advantages of sintered glass filter. Another physical method of sterilization you can see here, radiation sterilization, or you can simply say irradiation. Exposure of microorganisms to certain radiations. So usually they include electromagnetic radiations. The target of these radiations is nucleic acids, or target of these radiations is DNA and RNA. So when you expose any microorganisms to radiations, these radiations fall on the nucleic acid. Rather, these radiations are absorbed by nucleic acid, that is DNA and RNA, and damage occurs to these DNA and RNA. These radiations include gamma rays, UV rays also. Even these radiations can be very harmful to human beings. For example, UV rays or X-rays, because directly they fall on the DNA. Nucleic acids absorb, the bases absorb these radiations and results in mutations. One of the method of radiation sterilization is sterilization by non-ionizing radiations, and best example of these non-ionizing radiations is ultraviolet radiations. So these non-ionizing waves have very little penetration power. They range from 200 to 400 nanometer. So only microorganisms on the surface are killed. They have very low penetration power. They cannot travel to a very long distance. And upon exposure, these waves are absorbed by many materials, particularly nucleic acids. So when these UV radiations are absorbed by the nucleic acid, it results in formation of thymine dimer, or it is called pyrimidine dimers. There occurs unusual bond formation between adjacent thymine dimers, which bring about replication, which brings about errors in DNA replication and cause death of the microbes by mutation. This is the target of UV radiations, but the most disadvantageous thing about UV radiations is they have very poor penetration. They cannot travel to a very long distance. So formation of thymine dimers, which results in death of the microorganisms, is the ultimate effect of exposure of microbes to UV light. Then ionizing radiations. Examples of ionizing radiations include X-rays and gamma rays. These are very high energy radiations as compared to UV radiations. These radiations are also absorbed by nucleic acids, DNA and RNA, and result in ionization. So it results in many toxic metabolites, hydroxyl radical, superoxide ions, even ionization of nucleic acids. These are the ultimate effects of ionizing radiations. So with ionizing radiations, microbial resistance decreases with the presence of moisture or dissolved oxygen. Whenever there is a presence of moisture or dissolved oxygen, the effect of ionizing radiations decreases. Even temperature affects the effect of ionizing radiations. So both these ionizing as well as non-ionizing radiations can be harmful to human beings also, because they have similar effect on human cell or any eukaryotic cell. So these are the physical methods of sterilization. Firstly, we discuss methods of heat sterilization, then filtration sterilization and radiation sterilization. All these are physical methods of sterilization.